Good evening everyone. In today's video, we'll dive into a study about Staphylococcus aureus, a bacteria that is both fascinating and concerning in the world of food microbiology. I'm IQHWAN and I will be the first presenter for this topic. Before we move on deeper into the topic, what is exactly Staphylococcus aureus? Staphylococcus aureus is a gram-positive bacterium with a thick cell wall. It is spherical in shape and measuring about 0.5 to 1 micrometer. Under microscope, it is arranged in grade-like cluster. It also doesn't move or form spores but survive in microcolonies. Its cell wall made of thick peptidoglycan layer which help it resist physical stress. This bacterium also have thick acid which help it stick to the surface and evade our immune system. Some strains have a capsule or slime-like layer which protect them from desiccation and phagocytosis. And here's the kicker: as aureus produce protein A surface protein that interfere with ozonization. Next, this bacterium is also highly adaptable. It is grow best at body temperature, which is 37 degrees Celsius, but can survive in a range 7 degrees Celsius to 48.5 degrees Celsius. It also tolerates high salt level, low water activity, and even acidic or alkaline environment. Plus, it is also facultative anaerobe, a bacteria that can grow with or without oxygen, making it incredibly versatile in various food conditions. Next, for the survival mechanism, as aureus also produce heat stable toxins that are not destroyed during cooking or digestion. It also forms biofilm on surface like food processing equipment, making it hard to remove. Some strains are resistant to antibiotics and they produce stress protein that help it survive extreme conditions such as high heat and dryness. This bacterium is also very common. Up to 30% of people carry it on their skin or in their nose without symptoms. It spread through direct contact, contaminated surface, or improperly stored food. Food high in protein like meat or dairy are particularly at risk. If contaminated food is consumed, symptoms like nausea, vomiting, and stomach cramp can appear within a few hours.
I am SYAZLIANA and I will explain about assessing food safety risks of suspected item which is satay, nasi lemak, ayam goreng berempah, kerang rebus, cendol and fresh foods. First, for satay is at high risk of contamination with Staphylococcus aureus because it involves handling raw meat and preparing peanut sauce. If not handled properly, both can create ideal condition for bacterial growth. Raw meat can be contaminated with Staphylococcus aureus during slaughter, transportation, or preparation. Undercooked satay skewers that allow bacteria to survive. The internal temperature of the meat may not reach the level required to kill Staphylococcus aureus. Peanut sauce is nutrient-dense and protein-rich. If the sauce sits out at room temperature for too long, Staphylococcus aureus can produce toxins that are destroyed even if the sauce is reheated. Next is nasi lemak. Nasi lemak can easily become contaminated with Staphylococcus aureus because its key ingredients are highly perishable and require a lot of handling during preparation. Poor storage or hygiene practices can lead to bacterial growth and toxin production. Sambal made with chilies, garlic and onions is naturally acidic, but the acidity might not be enough to stop bacteria from growing if it's not stored properly. Boiled eggs also require peeling, which involve handling. If hygiene is up to standard, they can get contaminated, and peeled eggs left unventilated can quickly become a breeding ground for bacteria. Next, ayam goreng berempah. Ayam goreng berempah is highly susceptible to Staphylococcus aureus contamination because of the handling of raw chickens and the risk associated with proper cooking of storage. Since chicken naturally carries bacteria, poor hygiene or food handling can easily lead to contamination and illness. Raw chicken often carries bacteria like Staphylococcus aureus, which can easily spread to surface, utensils, and hands during preparations. Contamination can happen if raw chickens come into contact with ready-to-eat foods or cooked chicken through shell cutting boards, knife, or storage containers. Next, cendol. Cendol is highly prone to Staphylococcus aureus contamination because of its perishable ingredients, especially coconut milk, and the risk of poor hygiene during preparation. Since desserts like cendol are often left out for long periods, they can create the perfect condition for bacterial growth. Coconut milk is both nutrient-rich and highly perishable. If it's left at room temperature for too long, it can quickly foster bacterial growth and lead to toxin production. Shaved ice can become contaminated if it's made with unsafe water or handling using unsanitized equipment. Ice that comes into contact with dirty hands or utensils adds another layer of risk. Last but not least, these are summary that we can conclude based on their relevance to Staphylococcus aureus. For satay, nasi lemak, ayam goreng berempah, and cendol, they are highly prone to the Staphylococcus aureus. While for kerang rebus and fresh cut foods, we can call it as moderate to high risk of the contamination of the Staphylococcus aureus.
Assalamualaikum and a very good evening. My name is AIZAT with TERMIZI and I will be presenting on identifying common sources or reservoirs. So the first sources or reservoirs is the way of someone handling food. So basically, first point is improper food handling. Us as a human carriers, so Staphylococcus aureus is naturally present on skin, nasal passages, and tracts of about 20 to 30% of healthy individuals. So food handlers who carry the bacteria are one of the common sources of contamination. So the example of poor handling practices including not washing hands after coughing, sneezing, or touching their face, and preparing food while having infected cuts, wound, or skin infection, which can release large amount of Staphylococcus aureus into the food. So the next point is cross-contamination. From contaminated utensil and surfaces, for instance, when we use unwashed cutting boards, knives, or serving trays, can transfer Staphylococcus aureus from raw ingredients like raw chicken to ready-to-eat items like fruits or cooked satay. And that is shared equipment like skewers or grilling utensil, for instance, satay, may harbor bacteria if not properly sanitized between users. And the next thing is how cross-contamination can contact with, the bacteria can cross-contact with raw ingredients. For example, if raw chicken is marinated in a container and the same container is reused back to cook chicken without washing, so thus the Staphylococcus aureus can be transferred. Moving on to my last point, which is environmental factors. So I'll be talking about temperature abuse. Staphylococcus aureus grows rapidly in the temperature range known as the danger zone between 4 degrees Celsius and 60 degrees Celsius. Food left at room temperature for more than 2 hours can provide ideal condition for bacterial multiplication and toxin production. Example, in festival, for instance, Hari Raya, satay peanut sauce kept at room temperature for too long. And even cooked food like nasi lemak or ayam goreng berempah not held at enough high temperature could get the risk for the Staphylococcus to be rapidly grow inside or on it. And the last example is that cendol toppings, for instance coconut milk, that left unrefrigerated during the event will promote the growth of Staphylococcus.
Hi everyone, my name is Jordan. I'm gonna talking about the testing protocol for detection and isolation of Staphylococcus aureus from the foods by using the ISO method. So for the first test sample preparation, where in satay, we will extract the solid food samples such as the satay meat, sliced cucumber, onion, and curd rice. And the extracted liquid food sample can be the peanut sauce and marinade used for satay. And the extracted environmental sample can be the skewers used on satay and also the environmental swabs. The environmental swabs in this case indicates the swabbing or hand contact surfaces such as the utensil used for the foods. And for nasi lemak, the extracted solid food sample can be the rice, peanuts, anchovies, egg yolk, egg white, and also cucumbers. Liquid food sample can be the sambal and coconut milk used, and environmental sample can be the pandan leaf, banana leaf used for wrapping the nasi lemak, and also environmental swabs. So for the ayam goreng berempah, the solid food sample can be the fried chicken and kaffir leaves. Liquid food sample can be the spice marinade or sauce used, and the environmental sample can be the environmental swabs also. And the current rabus, the solid food sample can be the extracted boiled coconut meat. Liquid food sample can be the sauce used, and the environmental sample can be the environmental swabs. For the sando, solid food sample can be the pandan jelly, liquid food sample can be the chef ice, coconut milk, and palm sugar syrup, and the environmental sample can be the environmental swabs. Fresh cut fruits will be the solid food sample and also environmental swabs. So for the extracted solid food samples, we will weigh 25 grams of the samples aseptically and place into a separate and labeled storage bag. Whereas for extracted liquid food samples, we will measure 25 milliliters of a liquid sample and add into the storage bag. And then for both solid and liquid food samples, we'll add 25 milliliters of sterile BPW solution and homogenize in a storage bag. And then we undergo the incubation process and undergo the dilution process for further analysis. And for the environmental samples, we swab 10 cm square of targeted surface using a sterile swab, moistened with PPW solution, and place it into the 10 milliliters of PPW and vortexing for 30 seconds via the vortex mission. And we will undergo the same incubation process and dilution process as the above. So the next we will proceed to selective enrichment for Staphylococcus aureus from the sample by using the magnetic salt broth (MSB). So MSB is especially effective for selectively enriching S aureus from complex sample that may contain a wide range of microorganisms. The key components include beef extracts, mannitol, sodium chloride, and also phenolate, where the procedure includes transferring from the previous sample solution into MSB and then undergo the incubation process. So for Coccus positive Staphylococcus species, such as Staphylococcus aureus in this case, they will form mannitol producing acid which turns the medium from red to yellow, whereas for other species, which is non-Staphylococcus species and Coccus negative species, they will not form mannitol, so the medium will remain cigarette color. So let's move to selective plating. For backpacker agar, it's isolating and enumerating Coccus positive Staphylococci, which including S aureus in the foods, where the key components include the glycine, lithium chloride, potassium telluride, and your emulsion, and also sodium pyruvate. And for the procedure, we will split from the previous sample into the egg uptake and also undergo the incubation process. So for Staphylococcus aureus, which is complex positive, they will show a break grass shiny colonies with clear zones around the colonies due to proteolysis of egg yolk by the Staphylococcus aureus enzymes. And for the Staphylococcus intermediates, complex positive, which is Staphylococcus species besides Staphylococcus aureus, they will show a break grass shiny colonies with no clear zone of very weak reaction. Whereas for Coccus negative Staphylococcus species, such as Staphylococcus epidermidis and Staphylococcus prophyticus, they form a smaller colonies, often gray or off-white color, with no clear zone of proteolysis around the colonies. However, some species may form the opaque halos. So the final will be the biochemical test, whereas for catalyst tests, they are useful in differentiate between gram-positive foci, particularly Staphylococcus and Micrococcus species, which is catalyst positive, from Streptococcus and Enterococcus species, which is catalyst negative. And the key components include hydrogen peroxide and catalyst enzyme, where the procedure includes the isolating of colonies from the previous selective plating into a clean glass slide and adding with hydrogen peroxide to observe the reaction. And for final Coagulase test is distinguish Staphylococcus aureus from other Staphylococcus species, such as Staphylococcus epidermidis, which do not produce coagulase. The key components include saturated rabbit plasma and coagulase, where we will place and pick a well-isolated colony from the previous sample, the selective plating, into a glass tube, which containing the saturated rabbit plasma, and undergo the incubation process to observe the reactions. And for the catalase test, the positive result, Staphylococcus aureus will catalyze the enzymes and break down the hydrogen peroxide, producing oxygen gas which will form in the visible bubbles. Whereas for Coagulase test positive result, S. aureus will have a clot formation observed in plasma, indicating the presence of a free coagulase. So for the comparison of standard protocols, ISO-BIN for pathogens detection, ISO 6881 focus on backpacker agar with yolk with the typical colonies conformation with coagulase test and other biochemical tests. And for ISO 6882, they incorporate the MPM method for the sample flow contamination levels. As from a study from a published paper, they use backpacker agar effectively isolating S. aureus colonies with blackberry colonies and clear egg white reaction with coagulase test confirming the 96% specificity in pathogen detection. And for the BAM, bacteriological and organic manual, they emphasize using the metal salt agar or backpacker agar for isolation of Staphylococcus aureus, where they were subjected to further coagulase and catalase tests for confirmation. And the manual also recommendation of PCR for specific identification by focusing on the NUC gene, which is a nucleus gene. And a study from published paper indicates they have higher sensitivity, 99%, detecting S. aureus from the raw meat when supplemented with the PCR testing. So that's all from me.